Each patient is put into their own station in the andrology lab. The sample cup, paperwork, and tubes are all labeled with the patient's name and unique identifiers. Each patient stays within their own sample prep station. The patient's sample is sucked into a pipette. Volume is then measured and a small portion of the sample is brought to the macler for counting. A sperm counting device, known as a macler, is loaded with a very small portion of the semen sample. The andrologist looks at the macler under the scope and is able to obtain a sperm count based on how many sperm they see on the grid of the macler. The raw semen sample is overlaid on the gradient, a media that acts as a filter for non-motile sperm and debris. After the semen sample is overlaid on the gradient, the tubes are put into the centrifuge and spun at 1,300 RPM for 10 minutes. Once the gradient tubes are done spinning in the centrifuge, pellets can be observed at the very bottom of the tubes. This pellet contains a higher percentage of motile sperm. The pellet at the bottom of the centrifuge tube is removed with a pipette and moved to a pellet tube. Fertilization media is then added to the pellet tube and mixed. This pellet tube is placed into the centrifuge and spun at 800 RPM for 10 minutes. After the proper time has elapsed, the pellet tube is removed. The media on top of the pellet, called the supernatant, is removed from the pellet tube. The pellet is then resuspended in fertilization media in order to achieve the correct sperm concentration. To obtain a final post-processing sperm count, the macler is once more loaded with a small portion of the processed sperm sample. The processed sample is much cleaner and has a higher percentage of motile sperm.